Today we are going to demonstrate how to perform the A² multiplex immunoassay. You will need the following materials and equipment shown here, as well as the A² microarray system. After you have assembled all the items for the experiment, it is important to bring the A² plate to room temperature before removing the foil pouch. The first step in the assay development is crucial. The wells of the plate must be rehydrated with wash buffer. Using a multi-channel pipetter, deliver 100 microliters to each well. Incubate the plate on the bench top for approximately 5 minutes. Then remove the liquid from the plate, either by aspiration from the corner of each well using a pipetter, or by a rapid shake of the plate upside down over a sink. Do not attempt to dry the washed plate, since we'll be using it right away. For the purpose of this demonstration, we have serially diluted buffer with red dye to represent a series of standards. It is recommended that you prepare the standard dilutions, as we have done, in separate tubes rather than serially diluting from the plate wells. This will avoid scratching the well bottoms that contain the antibody array. Now, pipette 70 microliters of each of the standards into the first 2 columns of the plate. It is recommended to start at the bottom of the plate, wells H, with the reagent blank buffer, and then proceed up the plate with increasing concentrations of the standards as shown. The remaining 80 wells of the plate are reserved for your samples. Here they are represented using green dye. After you've filled the plate with all of your samples, replace the lid and mount the plate onto a platform shaker. Set the shaker on a low to moderate speed. Shake the plate for 1 hour. Remove the plate from the shaker. Remove the liquid from the plate, being careful not to touch the bottom of the wells with the pipette tip. The fastest and safest way to remove liquid is to invert the plate over a sink and shake rapidly. Now, rinse the plate with 100 microliters of wash buffer. Repeat the rinse process at least 3 times. Place the rinse plate back on the bench, but do not allow the plate to dry out. The next step is to add the biotinylated secondary antibody mix to each well of the plate. Pipette 55 microliters of the mixture to each well. Place the lid back on the plate and return it to the shaker. Incubate the plate while shaking as before for 1 hour. Remove the plate from the shaker and take off the lid. As in previous steps, carefully move the liquid from the plate wells. Wash the plate 3 times with 100 microliters of wash buffer. Place the wash plate back on the bench and proceed to the next step. In the following steps, we will dispense the signaling reagent, strepavidin, PBXL, to the plate. Pipette 55 microliters of the PBXL solution, represented by the light pink solution, into each well. After adding PBXL to all the wells, place the lid back onto the plate and return the plate to the shaker. Shake the plate for 1 hour. Remove the plate from the shaker. Remove the liquid from the plate. Pipette wash buffer into each well. Wash the plate as before for 3 times. Return the wash plate to the bench. Now add 100 microliters of the wash buffer to each well. The plate is ready to read on the A² microarray system. The A² reader will read each well of the plate. The integrated software program controls the reader, captures the well images, processes the raw data, and reports the results. We have opened the screen to manual mode to control the reader. Place the cursor over the appropriate window icon to open the door. Now, remove the lid from the A² plate and place in the holder on the reader's stage. Close the door and review the images as they appear on the screen. You may now open the door and remove the plate. The images have been taken and the comprehensive reports generated in under 5 minutes. At the heart of the Quanta Scientific's Oligo linkage system is a universal capture technology licensed from Beckman Coulter. The universal capture technology can be used to create tests for cells, DNA, RNA, and proteins. The most significant application of this technology will be an A² plate. The A² plate will increase the throughput of testing by increasing the density of tests in currently available testing platforms. Specifically, a 96-well micro-titer plate run on liquid handling robotic workstations. With the universal capture technology, Quanta Scientifics can place several hundred tests in each of the 96 wells. Suddenly, a 96-well plate can give researchers thousands of test results on a fully automated system, dramatically speeding results and reducing costs. The technology takes advantage of the specific binding between two complementary strands of DNA. One of the strands is placed at a specific location on a micro-titer plate to act as a glue to capture specific analyte assays of interest within the well. For immunoassays, the complementary DNA strand is attached to an antibody that will track the target analyte. Then the test sample is added and the analytes bind to their specific antibody. Next, other specific antibodies with fluorescent reporter molecules bind to the captured analytes. What is so unique about the A² technology is the ability to identify a number of different analytes all in one small well. Based on the amount of fluorescence, the concentration of the analyte can also be determined. This new technology has far-reaching applications, first in drug discovery and development, but eventually in specialty and patient testing. The Quanta Scientific system includes a 96-well array linker plate, a conjugation kit, buffers, controls, a signal development kit, a plate reader, computer, and image analysis software.